വെബിനാർ പരമ്പരയുടെ 20ാമത്തെ പ്രഭാഷണമാണ് ഇന്ന് നടക്കുന്നത്. ഏറ്റവും പ്രസക്തമായിട്ടുള്ള ഒരു സബ്ജക്റ്റാണ് ഇന്നിവിടെ ചർച്ചയ്ക്ക് വിധേയമാക്കുന്നത്. ഇത് നമ്മുടെ അതിഥിയായി എത്തുന്നത് ക്ഷത്രിമായം ബിർള സിംഗ് ആണ്. മണിപ്പൂർ യൂണിവേഴ്സിറ്റിയിലെ അസോസിയേറ്റ് പ്രൊഫസറാണ്. മികച്ച പ്രതിഭാശാലികളെ നമ്മുടെ കോളേജിന്റെ വേദികളിലേക്ക് കൊണ്ടുവരിക, പരമാവധി വിദ്യാർത്ഥികൾക്കും അധ്യാപകർക്കും പൊതുജനങ്ങൾക്കും അവരുടെ ദൈക്ഷണിക വ്യക്തിത്വത്തെ പരിചയപ്പെടുത്തുക തുടങ്ങിയിട്ടുള്ള ഉന്നതമായ ലക്ഷ്യങ്ങളാണ് ഈ വെബിനാറിന് പിന്നിൽ ഉള്ളത്. നല്ല ഭക്ഷണം, നല്ല വെള്ളം, നല്ല ശുചിത്വം, നല്ല ആരോഗ്യബോധം തുടങ്ങിയവയാണ് നമ്മുടെ ജീവനെ നിശ്ചയിക്കുന്ന മുഖ്യ ഘടകങ്ങൾ. പക്ഷേ നമുക്കറിയാം, ഇന്ന് ജീവിത ശൈലി രോഗങ്ങളാണ് 50% മരണങ്ങൾക്കും കാരണമായി തീരുന്നത്. ഇന്ത്യയിൽ ഏറ്റവും കൂടുതൽ ഡയബറ്റിക്സും ഹൈപ്പർ ടെൻഷനും ഹൃദയ സംബന്ധമായിട്ടുള്ള രോഗങ്ങളും ഏറ്റവും കൂടുതലുള്ളത് കേരളത്തിൽ ആണ്. കേരളീയരുടെ ഉയർന്ന ആരോഗ്യ ഉത്കണ്ഠയാണ് മരുന്ന് കുത്തക കമ്പനികൾ ബോധപൂർവം ചൂഷണം ചെയ്യുകയും അനാവശ്യമായ ടോണിക്കുകളും അപകടകാരികളായ നിരോധിത മരുന്നുകളുമെല്ലാം ഇവിടെ വ്യാപകമാക്കുകയും ചെയ്തത്. നോക്കൂ, ഇന്ത്യക്കാരുടെ വരുമാനത്തിന്റെ 40% ഭക്ഷ്യാവശ്യത്തിന് വേണ്ടിയിട്ടാണ് അവർ നീക്കിവെക്കുന്നത്. പക്ഷെ നമുക്ക് കിട്ടുന്നതാകട്ടെ മായം ചേർത്ത ഭക്ഷ്യോൽപ്പന്ന വിപണിയുമാണ്. അവരിൽ നിന്നാണ്. വാസ്തവത്തിൽ രോഗം മനുഷ്യാവസ്ഥയുടെ അടിസ്ഥാനപരമായ ഒരു സ്വഭാവമാണ്. അപ്പൊ അതിനെ മറികടക്കാൻ നമുക്ക് എന്ത് ചെയ്യാൻ കഴിയും എന്നുള്ളതിൻ്റെ ഒരു ഫലപ്രാപ്തിയാണ് ഇന്ന് നടക്കാൻ പോകുന്ന സെമിനാർ എന്ന് ഞാൻ വിചാരിക്കുകയാണ്. പല ഘടകങ്ങൾ, തൊഴിൽപരമായ, ദേശപരമായ, കാലപരമായ പല ഘടകങ്ങളും നമ്മുടെ ജീവിതശൈലി രോഗങ്ങളെ ബാധിക്കുന്നുണ്ട്, അല്ലെങ്കിൽ അതിന് കാരണമായി തീരുന്നുണ്ട്. നോക്കൂ, ഉഷ്ണകാലത്ത് ശീതവീര്യമുള്ള പദാർത്ഥങ്ങൾ ഉപയോഗിക്കുന്നതിന് വേണ്ടിയായിരിക്കണം നമ്മുടെ ഈ കാലഘട്ടത്തിൽ ചക്കയും മാങ്ങയും എല്ലാം ധാരാളമായി പ്രകൃതി തരുന്നത് എന്നുള്ളതാണ്. അപ്പോൾ ഇതെല്ലാം ഒരു ഭക്ഷണ സംസ്കാരത്തെയും ഒരു ജീവിത സംസ്കാരത്തെയും കുറിച്ചുള്ള നല്ല ചിന്തകൾ ഉണ്ടാകട്ടെ എന്ന് മാത്രം പറഞ്ഞുകൊണ്ട് ഞാൻ ക്ഷത്രിമായം ബിർള സിംഗിനെ കൊല്ലം ശ്രീനാരായണ കോളേജിലേക്ക് ഒരിക്കൽ കൂടി സ്വാഗതം ചെയ്തുകൊണ്ടും ഞാൻ നിർത്തുന്നു. നന്ദി, നമസ്കാരം.
Thank you, Sunil sir. Now I invite Dr. Shija UV for introducing our speaker.
Thank you, Disha teacher. I find immense pleasure in introducing our honorable guest, Dr. Kshetrimayum Birla Singh, who is presently working as Associate Professor in the Department of Zoology, School of Life Sciences, Manipur University, Canchipur, India. Before joining Manipur University, he has worked at Mizoram University, Azawad, as well as Sikkim University, Gangtok. He earned his Master of Science as well as PhD degree from the Department of Zoology, Center for Advanced Study, Punjab University, Chandigarh, India. He did his postdoctoral research at King's College, University of London. He is also recipient of Best Researcher Award at International Scientist Awards 2020 on Engineering, Science and Medicine by VDGOT Technology, Trichy, India, as well as Research Excellence Award by Institute of Scholar, Bangalore. His field of specialization is animal physiology and biochemistry. He has more than 14 years of teaching and research experiences. His areas of research interest are trace mineral nutrition research, diabetes mellitus, metabolic syndrome X, gene polymorphism in diseases, and recently, natural product studies and alternate medicines. He has to his credit altogether 40 research papers, books and book chapters in the journals and publishers of international and national reputes. He has also reviewed more than 22 research papers from the academic journals of international repute. At present, acting as an academic editor of 5 journals of international repute. Dr. Singh has also presented 35 research papers in many national conferences in India and in international conferences abroad. He has also delivered 10 invited talks as a research person. 1 PhD scholar has awarded under his supervision, and 4 students are currently undergoing PhD program. Dr. Singh, as principal investigator, has completed 6 research projects from different funding agencies like DST-SERV, New Delhi, BBT, GOI, and UGC. He has also organized more than 11 international as well as national level conference, workshops, and webinars as full-time coordinator. He is currently life member of 5 professional academic bodies in India. We are indeed extremely delighted to have you here today as a resource person. Over to you, sir. Birla Singh, sir.
Am I audible, madam?
Yes, sir.
Okay. So thank you so much, Dr. Shija, for formally introducing me to all attendees. Once again, a warm welcome and a very good morning to one and all. I hope at present you are staying at home, keeping yourself healthy and safe from this COVID-19 pandemic. As you know that we are now under the health crisis because of this second wave of the COVID-19, and it is utmost important and the huge responsibility for us to follow the specific guidelines which is issued by the concerned health departments of the states and the government of India to protect ourselves from the COVID-19 infection. And I also would like to encourage all the young students friends who are above 18 years of age to enroll themselves for the COVID-19 vaccination and protect from this deadly virus. At the outset, I would like to thank respected Principal Sir, Dr. R. Sunil Kumar, and the other key organizing member of the Agora 21 of the Sri Narayan College, Kollam, Kerala, for giving me this wonderful platform to share some of my research interest and research finding to the young students friends especially. I have been following this Agora 21 since last few days, and some of the talks are very, very interesting and knowledge enriching. And I consider myself very fortunate to be associated with Agora 2021, which is organized by one of the oldest and one of the premier colleges in Kerala, SN College, Kollam, Kerala, which is established in the year 1948, just after the India got independence. And I have visited the website of the college, and it's a it have a very beautiful campus, and the kind of research activities and academic activities which is undergoing in the college is very remarkable and commendable. And I have been observing that the respected principal, Dr. R. Sunil Kumar, also taking out time from his busy schedule just to present in the in this webinar and to deliver his principal remark. And I can say that under his able leadership, that college will be flourish as one of the best colleges, not only in Kerala but also all over the India. So with this introduction notes, let me start my lecture. So I'm presenting my Madam, can you see the presentation?
Yeah, it's perfect, sir.
Okay, thank you so much. So, dear friends, today I'm going to, discuss with you about the interesting talk on the title Excess Zinc in Diet and the Lifestyle Diseases. And here I'm going to discuss about how the zinc, which is one of the essential trace metals in our body and which is present in every foods item which we are consuming, and how the consumption of this excess zinc uh, for in the diet for longer period of time may be the one of the reason for the etiologic factors and the pathophysiology of the some of the lifestyle diseases like obesity, diabetes mellitus, and hypertension especially. And the you know that most of the research which is now going on in India and the uh, uh, abroad, they are mainly emphasizing on the deficiency of zinc and how the zinc deficiency affect our overall growth, our immune system, and our the reproductive systems. And there are very few scientists who are working on the that anabolic aspect of the zinc in our body. And I should say that I'm very fortunate that I'm also one of the scientists who have a very keen interest on the anabolic role of the zinc in our body and how it relates to the development of the lifestyle diseases. So before I go on my main theme of the topic, so let me share you the institutions where I have been working, because most of the attendees uh, might not have idea about where the Manipur University is actually located. So this is the bird eye view of the Imphal city. Imphal city is the capital city of the Manipur. Manipur is one of the northeastern states of India, and this is the map, so in the Indian map showing, and Manipur is just at the corner of the northern states, just bordering the Myanmar, and you will see the Kerala, which is at the extreme southern of the India, and so it's very interesting to see that I'm sitting at one corner of India, and you people, mostly those attendees from the Kerala, are on the other side of India. So coming to the location of the Manipur University, so this is the Imphal city, and the Manipur University is located just 7 kilometers from the heart of the city, and this arrow mark area is the location southern part of the Imphal city where the Manipur University is located. So Manipur University, just a brief introduction about my institution. So it's a central university established on the 5th June 1980, and the university has been accredited A grade by NAG in the year 2016, and this year we are facing another NAG assessment. At present, the university have 37 departments, 6 schools of studies, and the 109 affiliated colleges, 2 affiliated medical colleges, and 1 constituent engineering college. And this is the campus pictures of the Manipur University. This is the department I have been working now. So this is Department of Life Science. Under this Department of Life Science, we have Department of Zoology and Department of Botany. So now coming to the main topic of today's talk. First, I will discuss, and I will speak about about the lifestyle diseases, a brief overview of the lifestyle diseases, so that those attendees who are from the social science background might also understand what is actually the lifestyle diseases. And I will try to generalize my talk, because I have seen some of the participants are from the other backgrounds. So I will try my best to simplify and generalize my talk, even though it's a very scientific lecture. So if we talk about the lifestyle diseases, these are the diseases that are caused partly by our unhealthy behavior and partly by other factors. So or unhealthy behaviors like smoking, drinking, and sedentary lifestyles, and the other factors include that the these genetic factors and environmental factors. So under the lifestyle diseases, we have the obesity, which is the gaining of the body weight, and the coronary artery disease, and the diabetes mellitus, which is related to our blood sugar level in our body, hypertension, which is constant increase in blood pressure in our body, and osteoporosis, which is the loss of the bone mass, and cancer, and it's uncontrolled growth of the cell. So out of the many lifestyle diseases, my interest is in obesity, diabetes mellitus, and hypertension. So in this slide, I am showing why we are so much concerned about the lifestyle diseases. So lifestyle diseases, it's initially we consider it a degenerative, non-communicable diseases, because these type of diseases are mostly found and occur in the old age generation. But why we are so much concerned is that the prevalence of the lifestyle diseases is increasing day by day in the young age population also. And the other concern for the lifestyle diseases is that the most of the premature death, not only in India but all over the world, is because of the lifestyle diseases. And we have seen in this the graph that the the cardiovascular diseases, diabetes, and cancer, they are contributing 60% of the premature death worldwide. And since someone who is suffering from the lifestyle diseases, they have to visit to the hospital, they have to visit to the physicians for the treatment of the lifestyle diseases. And because of this, we have to spend lots of money for visiting a hospital and for visiting the physicians. So altogether, around 9 trillion of the global economy is going to be spent in the next decades, and it's likely to be double because of the treatment of the lifestyle diseases. That's why we are so much concerned about the lifestyle diseases. So what is the situation and the condition in India while we talk about the lifestyle diseases? So this is the report before the COVID-19 pandemic start from the Wuhan, China. So in India, there is the transition from the infectious disease, which is caused by pathogens, to the lifestyle diseases. And many people, especially there is men and women, are suffering from the cancer, from diabetes, from obesity, and their number is increasing day by day, even in the rural India. So obviously, so this affect our economy, and because we are spending money for the treatment of the lifestyle diseases. Sorry. So while we talk about the lifestyle diseases, we have to know what is the metabolic syndrome X. This is considered one of the risk factors for the development or for the future development of the lifestyle diseases. So metabolic syndrome X, it's nothing. It's the presence of the cluster of the medical conditions at one time, like abdominal obesity. Abdominal obesity is the deposition of the more fats in the abdomen region, in the adipocytes of our belly region, and we have dyslipidemia. Dyslipidemia is a condition where you have the high triglycerides level in the blood and the high LDL cholesterol. LDL cholesterol is known as the bad cholesterol, and at the same time, if someone have the low HDL cholesterol, then at that time the condition is known as the dyslipidemia. Then we have the insulin resistance. Insulin resistance is a condition where our cell could not properly utilize the insulin which is secreted from the beta cell of the pancreas, and because of this resistance, the cell could not get the proper amount of the glucose for its physiological functions. And the hypertension I have already mentioned in my previous slide. Is the constant increase in the arterial blood pressure in our body, and the high stress hormone. There is the high cortisol level. So if someone is diagnosed with the three medical condition of these five, then we can say that she or he is suffering from the metabolic syndrome X. So and he or she is likely to develop to lifestyle diseases in the near future. So these are the medical parameters for the diagnosis of metabolic syndrome X, and the components are, first one is the abdominal obesity. If the men have the more than the 40 inches of waist circumference, and women have the more than the 35 inches of the waist circumference, sorry, and the triglyceride level is more than 150 milligram per deciliter, and the HDL cholesterol level is less than 50 milligram per deciliter in man and in women, and there is the increase in the blood pressure more than 130 by 85 millimeter mercury. Then the fasting glucose level is more than 110 milligram per deciliter. That we can say that the C or D is having the metabolic diagnosed with the metabolic syndrome X. So this is the slide showing the visible syndrome of the metabolic syndrome X. In case of the women, because of the central obesity, so their body shape they will have the pear body shape, and in case of men, because of the central abdominal obesity, they will have the apple shape body. And you might have seen in some people that there is the browning of the folds of skin around the neck region and also in the armpits. So these are some of the visible symptoms that he or she might have the metabolic syndrome X. Now, in the coming slides, I will be speaking about a brief overview of the lifestyle diseases, especially obesity, diabetes mellitus, and the hypertension, before we come to the role of the zinc in the development of these lifestyle diseases. So first one is the obesity. Obesity is considered as the epidemic of the 21st century, and the major reason for the obesity is there is the imbalance between the energy intake and its expenditure, and in addition to that, if there is a minor reason because of the genetics and the lack of physical activities like sedentary lifestyle. And nowadays there is a very interesting research is going on about the leptin resistance in the obese people. And leptin is one of the hormone which is playing an important role in energy expenditure in our body and the food consumption. So if someone have leptin resistance, there is the high chances of he or she is having developed suffering from the obesity. So this is the slide showing the evolution of the man. So we have been learning since our childhood that we are actually evolved from the chimpanzee like monkey, and our ancestors are chimpanzee like monkey. And if this trend of obesity and if this trend of body shape is going on for another millions of 2,000 millions of year or 500 millions of year, then obviously our body shape will be changed to like a pig. And here I would like to specifically mention about one of the theory proposed by one of the famous geneticists from the University of Georgia, Professor Eugene McCarthy. So he proposed that the man is actually the hybrid species of the female chimpanzee and the male pig. So at that time when he proposed this theory, there are hue and cry from scientific communities and other evolutionary scientists that the theory is wrong. So considering that the trend of our evolution, our bodies have at present generation, and considering the what will be developed in another hundred of years, then the might be his theory also be proved correct, and that we might be finally coming to the shape of our forefather, that is the male pig and the female chimpanzee, as per the theory proposed by the Professor Eugene McCarthy. And the obvious syndrome, so it's a common picture in the present generations. So there are many obvious syndrome because of sedentary lifestyle or the consumption of many more junk food, and the many children are suffering from the obesity. So obesity is a formidable challenge because every medical conditions related to the lifestyles are linked with the obesity, like type 2 diabetes mellitus, dyslipidemia, and the cancer and other reproductive disorders. So obesity is usually measured using the BMI. BMI is body mass index. So BMI is calculated using the a simple formula by dividing weight in kg of the person by the height in meter square. And if your BMI lies between the 25 to 29.9, so we can say that you are overweight. And if your BMI is more than the 30, then you're suffering from the obesity. So this is the study reported in the USA, and you will see here that the chances of mortality risks also increasing as we increase our BMI. So both in the men and women, in the men and women. So now India is the third in the global rank of the obesity of epidemiology, just after United States of America and China, and the more than 300 million men and women are suffering from the obesity, and some are very young as those who are more than 20 years and above. So this is another interesting reports coming in very recently after the COVID-19 pandemic. So there are new reports that the COVID-19 is more deadly in the obese people, even though their age is very young. This is the study reported by the University of Oxford, United Kingdom, and maybe the reason maybe the obese people, that in obese people there is the high expression of the angiotensin converting enzyme 2 receptor expression, and this is the receptor which is used by the COVID-19 for entry to our body. And the obese people mostly they are having the lots of chronic inflammations because of the productions of the inflammatory cytokines, and the immune system are already compromised in the obese people. So because of this already existing conditions, so if someone who is obese, even though they are young, if they are infected with the COVID-19, there is the high chances that they will be hospitalized or they will be die from COVID-19 infection. So this is the the newspaper reports in India and also in the USA. So which is reported very recently, is obviously adding to COVID-19 complication risks in young, and why COVID-19 is more deadly people with obesity, even if they are young. So we have to be very concerned about the being obese during this pandemic. So now coming to the diabetes mellitus, it's a a metabolic disease in which there is the high blood sugar levels for prolonged period of time. So you know that we have insulin hormone which is secreted from the beta cells of islets of Langerhans of pancreas, and because of some defects in our cells, the cell cannot properly use the insulin, and because of the non-functional of this cell, so the glucose could not enter to the cell for its physical requirement. That's why there is more circulating level of the glucose in our blood. So at that time we can say that someone is diagnosed with the diabetes mellitus. So there are three types of the diabetes mellitus: type 1 diabetes and type 2 diabetes, and another third one is the gestational diabetes. So type 2 diabetes contribute 90% of the diabetes mellitus, and under this type of diabetes, as I have already mentioned, because of the insulin resistance, our cell could not use the glucose properly. And this type of the diabetes is mostly suffered in the people who are above 40 years of age. But nowadays the type 2 diabetes is reported, found in many young age population. That's why there is the concern to the scientific community why this happened, and why people of the young age population are suddenly suffering from the lots of diabetes mellitus. And the gestational diabetes is the diabetes type of diabetes which is suffered which is found in the pregnant woman. But in this case of diabetes, that the once the child is delivered, then the normal glucose level found in the pregnant woman. So if your blood glucose level fasting blood glucose level is more than the 150 milligram per deciliter, then you are having the hyperglycemia. There is the more blood glucose concentration in your blood plasma. So if it is more than 150 milligram per deciliter, then you will be diagnosed with the diabetes. And if your blood glucose level is in the 180 milligram per deciliter, then the sugar, your glucose will start appearing in your urine, and you might have found that in the diabetic people, their urines are affected with the ants. That is because of the presence of the glucose in the urine. This is also another interesting report that the diabetes doubles the risk of cardiovascular disease. If someone is having the diabetes and at the same time having the cardiovascular diseases, then there is the likely high chances that he will die because of the the cardiovascular disease. So diabetes enhance the complications of the cardiovascular diseases, just like obesity enhance the the complications of the COVID-19 infection. And most of our body organs are affected because of the high blood glucose levels. Diabetic retinopathy is the blindness because of the high blood glucose level in the blood vessel which are supplying to our retina. And diabetic nephropathy, this is because of the high blood glucose level, there is the leakage of the tubulus in the kidney. And diabetic neuropathy, because of the high blood glucose level, there is no nerve cell which is supplying, especially lower part of the body, in the ankle, in the toe are affected, and we feel numbness and the pain in the lower part of the body. There is a diabetic neuropathy. So it means that most of the our body organs are affected because of the diabetes mellitus. So now India is known as the diabetes capital of the world, and the number of the people who are suffering from the diabetes have increased dramatically since last 2 decades. So and the in terms of number, India is just after the China, because we are not the number one in terms of number, because China has having more population than India. So that's maybe the reasons why we are second in number. But if we calculate it in terms of ratio, so India is the can be considered as the diabetes capital of the world. So and because of this sudden increase of the diabetes mellitus even in the young age population, so that's how our interest for the role of the diet to the which are consuming in the living population of India, and what are the chemical components which is playing etiologic factors for the development of this blood sugar level in our body is we found it very interesting, and we that's why we try to do some research on this aspect. The lastly, hypertension. Hypertension is known as the high blood pressure, and it's a long-term medical condition in which the blood pressure in the artery is elevated persistently. And the hypertension is two types. One is the primary or essential high blood pressure, which contribute around 90 to 95% of the increased blood pressure in the living population. So in this type of the blood pressure, we do not know the exact reason behind why our blood pressure is increased. So and there is another type of the hypertension. There is a secondary or non-essential high blood pressure, and which contribute around 5 to 10%. And in this type of blood pressure, increase, so we know that the exact reason it might be due to the kidney failure. It might be due to some accident which damage our brain, which might be due to some other condition which is affected our body. So we exactly know that because of these conditions, the blood pressure in our body increase. But in the first one, we do not know that the exact reason behind the increase in the blood pressure. So hypertension is considered first risk factor for the future development of the cardiovascular disease. So as we all know, what is the the normal range of the blood pressure in our body? So if we have systolic pressure 120 millimeter mercury, then and diastolic 80 millimeter mercury, which this is considered as the normal range. Well, someone who is having the stage 1 and stage 2 hypertension, is diagnosed, if they are diagnosed with the high blood pressure, there is the 140 to 159 and more than 159 millimeter mercury in the systolic, and the more than 100 millimeter mercury in the diastolic. So in India by 2025, around 213 million people will be suffer from the high blood pressure. There is the hypertension, and the here the major concern is that the even in the rural people, village people, so there is the increased prevalence of the hypertension, which is not commonly found in the old time. Just like the obesity, which is playing a very complicated role in the COVID-19 infection. So diabetes mellitus and hypertension are also considered one of the comorbidities for the COVID-19 infection, and the people who are already having a diabetes mellitus and hypertension are likely to be hospitalized and likely to be dead from the COVID-19 infection. So we have to be very careful of the diabetes mellitus and hypertension. So now in the coming slides, I'm going to speak about the the important role of the the zinc in the diet. If we consume in high amount for a longer period of time, how it can develop to the lifestyle diseases and the most of my studies about the anabolic role of the zinc to the pathophysiology of the lifestyle diseases is done in the animal models, and we have done some studies on human subject also, and we have already, communicated with the Indian Council of Medical Research for the clinical studies of the role of the zinc in the lifestyle diseases. And once this COVID-19 problem is subsided, we will soon start the clinical, studies of the excess amount of zinc in the diet, and it's how it related to the lifestyle diseases with our finding on the animal on the animal models. So metallomics is known as the study of the metal ions, and it's considered a fifth branch of the building blocks. We have that we have genes, then the genomes, then the study of genome is known as genomics, then protein and the proteome, then proteomics. So just like that, we have metals and the metallome and metallomics. So metallomics is one of the emerging and trust area of the research, which is currently doing research in many parts of the world about the role of the metals which play in the various physiological functions of our body. This is the table showing the composition of the human body, and you will see here that we have different elements like oxygen, carbon, hydrogen, and the trace metals like zinc, copper, magnesium, molybdenum. And you'll see here that oxygen, comprises around 65% of the particles in the of the element found in our body, whereas that less than 1% of the body element component in our body is contributed by the trace these metals. But that the role which is played by this metal, especially the trace metal in our body, is it's very enormous. And it's in every physiological aspects of our body is because of these trace metals, even though they have in a very few percentage in our body. This is the periodic system of the bioelements, and we have essential elements like zinc, copper, manganese, iron, cobalt, and we have also essential non-metal. Even though they are non-metals, they are essential in our body, like the carbon, nitrogen, and oxygen. Now coming to the zinc, zinc is is an essential micronutrient. It's considered as trace metal because our body needs in minute quantity, and it's present virtually in every tissues in our body, and is particularly important for the correct functioning of the immune system and growth and development. And the zinc is component of the more than 3,000 enzymes which is found in our body and which is operating in our body. So if we look at the zinc proteome in our body, as about every 10 protein of the human body is a zinc protein. Either they will be a part of their structure as a cofactor, or they will influence their catalytic action, or they will be they will influence their regulatory mechanism. So so zinc is a very, very essential micronutrients, and without the proper amount of zinc, our immune system and our enzymes could not work efficiently. So this is the letter which is released from the physics science of the President of the United States of America. So I just would like to show how important is zinc in our body. So last year in the month of October, President Donald Trump was infected with the COVID-19, and out of the many medication he took, so he also, you will see the red circle, the zinc. He also took the zinc as one of the supplements to enhance his immune system. I hope that the many people which is present today are also taking zinc supplements, and because of the physician instruction that we just to enhance the immune system of our body. So because of this, and requirement of the zinc in our body, so the recommended dietary allowance, which is led by the WHO Health Organization, is that starting from the 3 milligram in case of infant and children, so adults, adult male and female can take 20 milligram zinc per day. But in case of the pregnant women, so they have recommended 25 milligram of zinc per day for the fulfilling the various physiological functions of the zinc in our body. Now, what are the reasons for increasing the bioavailability of the zinc in our body? So that is our major concern. That during the last 2 decades, zinc as a zinc sulfate is being used, indiscriminately in the fertilizers, in agriculture and the animal husbandry practices. You might have seen that the chicken feed and the pig feed, even the feed which is given to the cow. So there is a lot of zinc sulfate. So because zinc is non-toxic and it's increased the body weight to the consumers. So because of this, so the animal husbandry farmers, they used to give lots of the feed which is having the greater amount of the zinc, and just to commercially exploitation of the animals, because if they feed lots of zinc, then the animal will grow faster and they can sell the chicken or the pig or the cow for the commercial purpose. So that's why the amount of the zinc in this animal tissue is increased manifold because of the indiscriminate utilization of the zinc sulfate, not only in the husbandry practices, even in the farmers also using fertilizer lots of zinc sulfate for the commercial for the faster growth of the plants, rice or whatever the plants which they are growing in the fields. So this increased the bioavailability of the zinc in the diet. And in addition to that, zinc is also used in baby food and multivitamin supplements. If you check on the baby food supplement which is available in the market, so there will be there will be zinc supplements that are written in the cover of the most of the food and vitamin supplements. So and this is one of the reasons why we are consuming lots of zinc for longer period of time. And nowadays most of us are consuming lots of junk foods, and these junk foods, there is no fibers and phytates, and we are avoiding most of the food items which have lots of phytates and fibers. So these phytate and fibers are very, very important in our body, because they they restricted the more consumption of the zinc in our body, especially through the small intestine absorptive cells. And if you are consuming more junk food, means you're likely to absorb more zinc in our food. So the consumption of junk food by children as well as by the adult men and women is one of the reasons for the increasing the zinc, a greater amount of zinc in our body. And we have done a study and a survey on the while I was working as a PhD scholar in the Punjab University, because we have surveyed all the vegetable items and with foodstuffs which is available in Punjab, and we found that the zinc level is more than 40 milligram zinc per kg and 120 milligram zinc per kg in the above ground and underground vegetables like carrot and the pumpkin and the gajar, like etc. And you will see that the daily recommended range is between the 12 to 20 milligram zinc per kg. But while we survey the amount of metals, the other metals like copper, magnesium, manganese are within the normal range. And there is another very interesting report from the United States of America, which is done by the Arsenal on ground in the year 2003. So they reported that the percentage of the zinc consumed from the zinc fortified foods, means in the affluent countries, that most of the breakfast they are provided with the fortified foods. So and here there is they added lots of zinc. So the amount of the zinc consumption from the zinc fortified foods double from the from 14% to 28% within a short span of year. That is the 1990, between the year 1994 to 1998 in the preschool US General, and this also they found in all other adult age group, and they reported that the percentage of this zinc consumption from the fortified foods will increase over time. So it means through the diet, even though the amount of the zinc in some of the vegetable or some of the meat food stuff might be below the permissible limit, but we are consuming more zinc and the more zinc through for a longer period of time. And we also done some of the the food items which is collected in the markets in the northern states of India, in the Mizoram, from Manipur, and in the most of the meat food stuff which is available, which is found in the markets, we found the zinc level more than the permissible limit, and while the other level of other metals are within the permissible limit. So now, what are the anabolic roles of the zinc in our bodies? This is very, very important, and because of these regions, so there is likely to develop to the the lifestyle diseases, complication of the metabolic syndrome X if we consume zinc through our diet for a longer period of time. So first is that zinc promotes absorption of nutrients. If we have the zinc in our diet, then there will be more absorption of fats, there will be more absorption of glucose, there will be more absorption of the proteins, and this have been reported by many scientists from the different parts of the world. And zinc induce the proliferations of the adipocytes. This is also a very interesting finding, that the adipocyte cells where our fats are stored, and which are commonly found in the belly, in the pelvic region of our body. So if there is the high presence of zinc in our body, then the premature adipocytes is likely to be changed to the mature adipocytes, and at the same time the population of the adipocytes will be increased over time. And the zinc is component of the insulin. This is the photographs on the right side, which is showing the pancreatic beta cells of the mouse, and the zinc insulin is stored in the form of the zinc crystal, and the zinc has the potential to mimic the insulin activity in our body. So because of these specific roles of the zinc in our body, that they might be playing an important role in the development of the these lifestyle diseases. What is the specific role of the zinc in the obesity? In the human being, especially during the growth period, zinc it activates the growth hormone level. So they activate one of the gene of the growth hormone, which is known as the ovine metallothionein growth hormone transgene, and it enhances the amount of the growth hormone concentration in our body. So because of the increased elevation of the growth hormone level, the insulin level also increase in our body, and the number of the adipocytes in our body also increase at the same time. And once there is the cessation of the growth period in the human being, the elevated level of the insulin will have more deposition of the extra glucose, which is absorbed because of the presence of the high zinc to the adipocyte cell. So there is likely to produce an observed state of the obesity in the coming years. So this is the one of the physiologic role which played by the the zinc in the obesity. So we also confirm it through the animal model studies. So what we have done is that we just to see the real life situation. So we divided the rats into different groups, group 1, group 2, and group 3. And in the group 1, we used to fat and the sugar-rich diet, having the amount of zinc which is permitted by the WHO. There is a 20 milligram zinc per kg diet, and in group 2 and group 3 we increase the amount of zinc to the 50 milligram per zinc, and to the 80 then in group 3 80 milligram zinc per kg diet. And we fed all this group of rats for another 180 days. There is for period of the 6 months. There is just to see just to compare the real life situation. So what we found is very interesting, that the body weight of the animals in the group 2 and group 3 rat is increased significantly as compared to the group 1 control. And while we check the urine glucose of these rats, and starting from the day 60, so the glucose in the urine of this rat start showing, and while we test with the Benedict test, we can then finally confirm that these rats are having the glucosuria, and they are diagnosed with the metabolic syndrome X. So this is the photomicrographs of the histochemical studies on the skin on the ventral portion of the abdomen of that 3 group of rats. And here you will see that the development of the adipocyte cells on the skin while we perform with the Sudan black bee methods. So you will see here in the first picture, there is the single layer of the adipocytes, which is represented by EA, and on the second picture you will see that there is the increase in the population of the adipocytes, which in the last picture you will see that the population of the adipocytes in the group 3, which is fed with the 80 milligram zinc per kg diet, is increased, and even they are extended to the dermis region of the skin. So this also corroborated the body weight, or the finding of the increase in the body weight. The adipocyte cells of the rat increase on the zinc dependent manner. We also studied the transmission electron microscope studies of the intestinal microvilli cell of the small intestine of the 3 group of rats. And here you will see that on the first one, there is the normal development of the microvilli, which is one of the important cell for the absorption of the nutrients through the small intestine. And you will see here in the group 2 and the group 3, which is represented by the picture number 2 and the 3, there is the microvilli is increased in the height and increase in the number. And even in the picture number 3, you will see that there is the height of the microvilli is increased further, and the population of the microvilli also increased. So this shows that the previous finding of the other scientists, and that the zinc presence of the consumption of the excess zinc through the diet increase the microvilli populations, and they increase the more absorption of the nutrients. So this might be one of the reason for the gain in the body weight in those group of rats. And we also done one study where we check the insulin leptin hormone level in this group of rats, and we found that there is the increased concentration of the leptin hormone level in this obese rat that is in group 3 and group 2, and we publish it in one of the good journal of medicine and biology, we also publish another paper in the Science and Technology Journal out of this finding. Further, while we check the blood pressure of those rats, which are fed on the high or greater amount of zinc for longer period of time, there is no increase in the systolic pressure of the rats very significantly as compared to the group 1 control. So this is another very interesting finding. And we also check the the blood profiles of the rats, and here you will see in this bar diagram that the concentration of the glucose is increased significantly in the group 2 and group 3 rats as compared to the group 1 control, which is fed on the normal level of the zinc, while the the level of the cholesterol, triglyceride, and LDL cholesterol is also increased significantly in the group of rats which are fed on the greater amount of zinc, and while the good cholesterol, there is HDL cholesterol level is down in this group 2 and group 3 rats as compared to the group 1 control. So it means that these rats are suffering or having diagnosed with the hyperglycemia and dyslipidemia. So that is the one of the condition of the metabolic syndrome X, and obviously these rats in the near future will likely to develop the most of the conditions of the the lifestyle diseases. We also checked in hormones levels in this different group of rats, and here also we found that the concentration of the insulin in the blood increased significantly in group 2 and the group 3 rats. Cortisol level, stress hormone level also increased in the group 2 and group 3 rats. Aldosterone level also increased in group 2 and group 3 rats, while the triiodothyronine, thyroid hormone level, T3, T4, and TSH is decreased in the zinc supplemented group as compared to the normal zinc supplemented group. So it shows that these rats are also having hyperinsulinemia, hypercortisolemia, hyperaldosterolemia, and they have very low basal metabolic rate. While we check the adrenal cortex using the electron microscope, and what we found is that there is the increased formation of the lipid globules in the group 2 in a group 2 rat as compared to the group 1 control. We might have seen in the first picture is the normal adrenal gland of the this group 1, and the second one is the adrenal cortex of the group 2, and third one is the adrenal cortex of the group 3, and you will see there is the suckle globules, lipid globules, and the number of the population of the lipid globules increase dramatically in the group 2 and group 3 rats. So it shows that these rats which are supplemented with the high zinc diet are under the in elevated steroidogenesis. There is the elevated for activities of the cholesterol and the steroid hormone formations. That's why there is the increase in the aldosterone level. That's why there is the increase in the in the cortisol level, and the further we also check whether the oxidative stress in these rats are actually compromised or not, and we selected some of the important antioxidant enzymes, and when we estimated, we found that the level of the this antioxidant enzyme like superoxide dismutase catalase, glutathione transferase, glutathione reduce, all are decreased significantly as compared to the group 1 control. So it shows that these rats are under the constant oxidative stress when we fed them with the greater amount of zinc for a longer period of time. This is also another interesting finding. There is the metal imbalance in the most of the tissues of these group 2 and group 3 rats, which are fed on the in a diet which is having greater amount of zinc. You will see here the serum zinc level is significantly high in group 2 and group 3 as compared to the group 1 control, while the level of the other essential metals like copper and magnesium is decrease in the here in the abdominal muscles and the other tissue like liver. So it shows that when we have the we supply with the rats with the greater amount of zinc, there is the metal imbalance. Might be because zinc have a antagonistic interactions at the intestinal absorption level with the copper and other metals. That's why that the zinc promotes the deficiency of the other essential metal like copper, magnesium, and the manganese. But the parametric data of the manganese, we have not shown it here. So this is another interesting finding from the electron microscope studies, and in the 3 group of rats. And you will see here that there is the reduction. This is the picture of the pancreas of the rats in the group 1, group 2, and group 3 rats. And you'll see here that there is a reduction in the zymogen granules in the group 2 and group 3 rats as compared to the group 1 control, and there is also increase in the dilatation and vascularization of the endoplasmic reticulum. And this shows that while we fed the zinc for a greater amount to the rats for longer period of time, there is the increased activity of the pancreas of the cell, and because of this overactivity, there is the exhaustion of the cell. We have done a human, based human subject studies in Chandigarh populations in collaboration of the Postgraduate Institute of Medical Sciences, PGI Chandigarh, and what we found is that very interesting that the amount of the zinc in the tissue, especially in the here in the nail of the human subject, which are suffering from the diabetes mellitus, which are suffering from the osteoarthritis, which are suffering from the obesity or hypertension, are more as compared to the normal human subject, which doesn't have any such lifestyle diseases. So it shows that since the population of this Chandigarh is consuming more amount of the zinc over a long period of time, so there is the more deposition of this zinc in the tissues of this population, while the other metals like copper, magnesium, and manganese are found in the normal level and comparable as compared to the control human subject. And another interesting finding is that those descendants child of the parents having the family history of the diabetes mellitus, obesity, and hypertension, even in those childs also, in those descendant also, there is the high level of the zinc deposition in their here in the nail. So this shows that some of the human populations which are genetically predisposed to retain and to absorb more amount of the zinc through the either through the diet and through the diet, especially someone if they are consuming more food stuffs which is having high concentration of zinc. So we have published our finding to the some of the reputed journals, and what we found is that what is the overall conclusion is that if we fed a greater amount of zinc through the diet, then they may induce obesity, they may induce glucosuria, presence of glucose in the urine, and they may induce the increase in blood pressure and they also produce hyperglycemia, increase in blood glucose level, dyslipidemia, increase triglyceride level, and decrease in the HDL cholesterol, increase in the insulin level, hyperinsulinemia, and increase in the stress hormone level. So they also induce oxidative stress in our body, as you have seen in the antioxidant enzyme activity status in the previous bar diagram, and they also cause ionic imbalance leading to the deficiency of copper, magnesium, and manganese. So these are some of the the important finding which out of our studies, and this might lead to the development of the obesity, diabetes mellitus, and the hypertension in the rats which are supplemented and which are fed on the greater amount of zinc through the diet. So with this backgrounds in my in my mind, so I thought that I study some of the in-depth research, molecular studies about the role of the zinc in the lifestyle diseases. So then I'm very lucky enough that I got the opportunity to work in one of the internationally acclaimed research project that is titled role of the zinc ion in the cell signaling and phosphorylation. That project is sponsored by the BB SRC United Kingdom, and I got under this project I got the opportunity to work in the laboratory of the Professor Wolfgang Murray of the King's College University of London, and who is the principal investigator of this project in collaboration with the other leading scientists from USA and Japan. So there I got the opportunity to work on this how the zinc modulates the insulin signaling and leptin signaling, and that might link to the pathophysiology of the diabetes mellitus and the obesity. So in the coming slide, I'm going to speak on how does zinc play an important role in the signaling of the insulin and leptin hormone, and I'm also going to show some of our findings out of that studies. So what are the recent studies which is coming on based on the zinc is that zinc is not an antioxidant. So previously what we consider is a zinc is an antioxidant, but that the deficiency and the excess of zinc in our body can cause many type of the abnormalities. So that's why the scientist has come to the know that zinc doesn't play any role in antioxidant, and the zinc is also redox inert in a biology. This is the recent finding, because zinc ion have the only +2 oxidation state, and they play a very important role to the redox regulations of the many enzymes, proteins of our body. So this is the photo slide showing the recent studies on the zinc biology, what popularly known as a zincology. So starting from the 1869 to 1963, most of the research related to zinc is on the nutritional aspect, means why zinc is essential in our body. And afterwards, then these are the name of the scientists, from David Kaelin to the Ivanovo Bertini. So they are working mostly on the zinc, how zinc play a role in the activities of the enzymes and other proteins, like how they act as cofactors of the many importantly physiologically important enzymes. And after 2006 onwards, zinc, most of the research related to the zincs are on the cellular signaling and how they control the signaling in our cell. So our project is part of the role of the zinc in the cellular signaling and its phosphorylation. So zinc ions complement the calcium signaling, because calcium is one element which is already known for the signaling functions in our body, and now the recent study has come up that the zinc also play an important role in the cellular signaling, and which complement the already existed function of the calcium ion. That one interesting aspect of our the aspect of zinc in the cell of our body is that the concentration of the zinc in our cell is tightly maintained by some of the transporters of the zinc, what we known as the zinc transporters. So these are the proteins known as GIF protein and ZNT protein. So there are two types of zinc transporter. One is ZNT4 T4 zinc, and another one is GIF transporter. So these two transporter actually working, in our cell to tightly maintain the amount of the zinc in our body. So because of this, tight control of the zinc transport of the zinc transporter proteins, so most of the zinc level in our body cell is maintained within this range. In case of the mitochondria, 0.1 to 300 picomolar. In case of endoplasmic reticulum, in the range of 1 picomolar to 5 nanomolar, and in case of cytosol, in the range of 0.4 to 1.5 nanomolar. So this is possible because of the various transporter which is present in the cell. Like zinc, other metals in our body also tightly controlled by the various exchange protein, the various transporters, and these are the cartoon picture of some of the transporter of our cell. Now protein tyrosine phosphatase 1b. So this after joining the Professor Wolfgang Maret laboratory, so my interest in order to look the relation of the zinc with the lifestyle diseases. So I selected this protein tyrosine phosphatase 1b, one of the the protein, one of the enzyme which acts as the negative regulator of insulin and leptin signaling pathway in our body, for my study under the insulin and leptin signaling project of the BBSRC United Kingdom, and we have found some very interesting finding out of this work, and before coming to my findings, so this protein PTP1B is as I just mentioned, is a negative regulator of insulin and leptin signaling pathway, and it is highly expressed in case of diabetes mellitus and in obesity and some form of the cancer. And it's a highly the location of this PTP1B protein tyrosine phosphatase is just at the cytosolic phase of the endoplasmic reticulum, and you'll see the picture on the cartoon picture on the lower side of the slide. The PTP1B is located and expressed in the endoplasmic reticulum. So this is the crystal structure of the PTP1B, and after coming out this crystal structure, we have come to know that the active site and signature motif of the PTP1B, and here the cysteine 215 played an important role for the for the activities of this PTP1B in insulin signaling. So what actually this PTP1B do in our cell signaling, especially in the insulin and the leptin signaling. So here you will see the cartoon picture of the insulin and the leptin signaling. Once the insulin bind to its receptor in the cell membranes, so there will be the phosphorylation of the tyrosine residues of the insulin receptor kinase. Okay, and the PTP1B, which is present in the endoplasmic reticulum, which is expressed in the endoplasmic reticulum, they will dephosphorylate. They will remove the phosphate group from the tyrosine residues of the IRK, and they will they will stop the insulin signaling. Because of this, break in the insulin signaling, the glucose gate could not be opened properly. Because of this, there will be insulin resistance, and glucose cannot be absorbed through the cell. Just like that, in the leptin signaling also, the leptin hormone once bind to the leptin receptor, it phosphorylates the tyrosine kinase protein of the JAK2 and JAK2, and this PTP1B, they immediately will phosphorylate the JAK kinase, and they will stop the downstream cascade of the reactions, and the leptin hormone will not do function properly because of this dephosphorylation. So then if we inhibit the activity of this PTP1B in the endoplasm, which is expressed in the endoplasmic reticulum, then there is the high chances that we might come out a novel drugs which can enhance the insulin and leptin signaling in our body, which is related to the the insulin resistance and the development of the obesity in our body. So there are many research which is going on to target this PTP1B to produce a therapeutic drugs, especially the metal-based drugs and the metal-based drugs, and to control or to modulate the activity of this PTP1B, which is acting as a brake in the insulin and leptin signaling. And one important aspect of this, physiological aspect of this PTP1B is that there is another zinc transporter. You will see here in this cartoon picture, GIP7. The GIP7 is the zinc transporter which import the zinc ion from within the endoplasmic reticulum to the cytosol of the cell, and because of this import of the this zinc ion from the endoplasmic reticulum to the cytoplasm, so the released zinc ion might influence the activity of the PTP1B. This is our interest, and we do the kinetic study of this PTP1B with using the different concentration of the free zinc ion in the body, and we found that this zinc ion can inhibit and they can modulate the functions of the PTP1B in the nanomolar range. So what we found is that the PTP1B activity was modulated or inhibited by the zinc ion in the range of the 2.4 nanomolar, and not only the zinc, other metal ions like the copper and the cadmium also can modulate the activity of the PTP1B in the range of 600 femtomolar and 7.3 nanomolar. And we also, in order to confirm further the structural binding between this, PTP1B and zinc ion, we do the molecular docking in collaboration with one of the scientists from Italy, and what we found is that this is the crystal structure of the PTP1B, and the gray color round ball represents the zinc ion, and we found that there is the binding side of the zinc to the active side of the PTP1B in the closed form of the PTP1B, and this we have already published in the some of the reputed journals. And now one thing which is limited to our study is that I could not able to do the actual amount of the zinc present in the cells of those rats. Those rats, after the supplementation of the high zinc through the diet for a longer period of time, because of the limitation of the time. But now I am also planning to collaborate with the London metallomic facility in order to study the metal concentration using very powerful sophisticated spectrophotometer that is known as the laser ablation inductively coupled plasma mass spectrometer. So using this spectrometry, we can see the the concentration of these metals in the living cells of the rats. So this is what we are planning, and then we after doing this study, we can confirm that after the supplementation of the high zinc, how much zinc we have in the pancreas or in the adrenal cortex or in the liver, and how they are going to influence the PTP1B, and which is playing an important role in the insulin and the leptin signaling. So that also we need to confirm in the coming days. And presently we in order to find more in-depth studies about the role of the zinc in the pathophysiology of the lifestyle diseases, we also have done the most of the work about the gene expression of some of the important antioxidant enzymes like zinc superoxide dismutase, catalase, and glutathione peroxidase in the rats. We have already, and the leptin receptor gene in the adipose tissue of the rat. Using the same animal models, using the same experimental design, we have already done the work, and only the analyzing part of the data is remain. So hopefully in the coming days we will come out another interesting finding. In the biochemical study, we have already found that the level of these important antioxidant enzymes activities decreased significantly in the zinc supplemented group. But we want to know on the molecular level, on the gene level, whether there is the upregulation or downregulation of the expression of these, the genes of these antioxidant enzymes in these rats. And another, interesting thing is that we also done some of the work on the first generation of the rats born from this group 2 and group 3 rats, which are supplied with the greater amount of zinc. As I have mentioned in the human subject study, that the child of those parents who have the family history of the diabetes mellitus, obesity, and hypertension also have the greater amount of the greater deposition of zinc in their hair and the nail tissue. So we would like to see in the animal model whether these are genetically linked, and whether there is a carryover effect of the greater amount of zinc to the next generation. So this is what another target to link with the next generation of the hazard effect of the zinc in our body. So some of our finding are selected as the one of the best research finding by the United Nations Library of Medicine, and we are selected one of the top second in the top 50 research publication in the year 2017 to 2012, and our research finding also highlighted in the website of the Food and Agriculture Organization of the United Nations. So after all this finding from the research, so I try to communicate with the common people of my state. So I wrote a journal article in the newspaper, daily newspaper, and even I published one small book in order to convey the message about the hazard effect of the consumption of the excess zinc through the diet for a longer period of time, and how it can develop to the lifestyle diseases. So I also have presented whatever the finding in some of the international conference abroad in the Bangkok in the year 2009, in Singapore 2012, and very recently in the University of Cambridge in the year 2015, and even I'm fortunate to get some of the one awards for the best one of the young scientists in the International Conference of Nutrition on Bangkok. So what are the take-home messages? So the rising prevalence of lifestyle diseases even in the youngest population during the past 2 decades may not be essentially due to the change in the lifestyle, because as I've told you that most of the lifestyle diseases are because of our unhealthy behavior like sedentary lifestyle, but also may be associated with the excess zinc intake through diet, because as I've told you, many people are genetically predisposed for the excessive absorption of the zinc through the food, and they are susceptible for the greater absorption of greater amount of zinc. So if they have the more absorption of zinc, then obviously there will be changes in the physiological functions in their body. So this more absorption of the zinc occurs through the food intake and through the food chain due to its liberal use of the as zinc sulfate in agriculture and animal husbandry practices employed by the farmers. As you know, after the green revolution in India, as I have told you, there is the liberal use of zinc sulfate in the animal husbandry as well as the agricultural practices by the farmer, just to exploit commercially the products of their farm, and this increased the more consumption of the zinc through diet. And nowadays there is a zinc fortified foods and the zinc rich dietary supplements, and most of our breakfast, which is like which is present, which is found in the markets, are richly supply enriched with the zinc concentration of zinc. And as I have told you that we have increased the consumption of the more junk food. Because of the more consumption of the junk food, we are taking less fibers and phytates in our diet, and this also enhance the more absorption of the zinc through our body, because this fiber and phytates play an important role in the limitations of the zinc absorption through the small intestine. And the zinc zinc ion, they have the potential to inhibit or modulate the PTP1B and nanomolar range, and this PTP1B is an important enzyme which play a negative regulator of the insulin and leptin signaling, and these two hormones are important hormone for the diabetes mellitus and development of obesity. But we are yet to confirm means how much zinc we can found in the cells after the high supplementation of zinc in the diet. So they are yet to confirm using some of the highly sophisticated techniques. So I have many people to acknowledge for my studies. Professor S.K. Taneja, he's my PhD supervisor in Punjab University, Chandigarh. Of course, the photograph is not very clear. He has already retired, and he is the UC Professor Emeritus. And now, and the next is the Professor Wolfgang Moret. He is also my mentor during my postdoctoral research. And the Professor Christer Hoogstrand, he is also my co-mentor in the University King College, University of London. Dr. Matthew Matt, who this guy who is standing here, he helped me in analyzing the kinetic study of the PTP1B, and some of my lab mates, Dr. Elisa from Italy, Dr. Sandra from Croatia, Portugal, Dr. Dolgas, now he's in the USA from the United Kingdom, and Dr. Seri Sasdeva, he's from Delhi, now settled in the USA. And the Professor Wolfgang also come down to the India in the year 2016, in the year 2017 in the Bangalore. There is a two international conference held consecutively 2 years for 2 years in 2016 and 2017 in the Hindustan Unilever Limited Research Wing, and he came down to deliver a keynote lecture, and I got the opportunity to meet him after my completion of my postdoctoral research. These are the photographs, and I would like to acknowledge all my funding agencies: University Grant Commission, DSD Science and Engineering Research Board, BBSRC Bioscience for the Future. This is the funding agency which is which sponsor my my postdoctoral research project. Department of Biotechnology, DBT, Ministry of Science and Technology, and the Royal Society of Chemistry in King's College, London. And also I would like to thanks respected principal Sir Dr. R. Sunil Kumar, principal of the Sri Narayan College, Kollam, Kerala. Dr. S. Jisa, coordinator of the this Agora lecture series. Dr. Shija, he's also one of the coordinator of the Sri Narayan College, Kollam, Kerala, and Dr. Rajas Selvam, who is the assistant professor working in the Vivekananda College, Tamil Nadu. Through her only, I know Dr. Shija and Dr. Jisa, and the and thank you so much all the attendees. And this is the two beautiful pictures of the one is animal, one is the flower. The animal is the peculiar of this animal and this flower is that the this is found in Manipur, and the name of this deer is known as the Cervus ildi in scientific, and locally Shanghai, and this is found in one of the national park of the Manipur and doesn't found anywhere. It's one of the endangered species, and the flower is known as lily. This is discovered by one of the British traveler, and this is also found in the only the Manipur and not in other parts of the world. So thank you so much everyone for your kind patience and listening, and thank you so much.